Hello everyone, I Assistant Professor V Shete from Department of Microbiology, KJ Somaya College. In this online lecture, we will study about the parasite, human parasite called as the Entamoeba histolytica. Okay. Now this Entamoeba histolytica is first described by the Lost in the 1875. Okay. After being isolated, and this was first isolated by him from the patient who was suffering from the dysenteric stools. All right, and this parasite or this protozoa will be responsible mainly for that conditions only. Now this is the trophozoite. Okay, you can see in that picture. Now it is up to 60 millimeter in the diameter. Okay, before that, this parasite exists in the three morphological forms. First is the trophozoite, precyst and the cyst. These are the three morphological forms that are present in this parasite. Okay, first one is the trophozoite forms. Now in the trophozoite, it is up to 60 μm in the diameter, endoplasm, granular endoplasm, then it will infect the RBCs, leukocyte and the tissue debris. That means it will invade in the food vacuoles. Okay, then it is motile, then blunt single pseudopodia, single large nucleus it has. You can see in that picture it is having the single large nucleus. Only trophozoite present in the tissues. This is the only trophozoite that is present in the tissues, right? After this, the basic classification of this protoparasite. Now phylum is the Sarcomastigophora, subphylum is Sarcotina, superclass is Rhizopoda, class is Lobosia, order of this class is Eumebia, genus is Entamoeba and the species is Histolytica. That is why we called it as Entamoeba histolytica. Okay. Now we saw first one, morphological first form, that is the trophozoite. Now second one is the precyst. Okay. Now in the precyst, you can see in this picture it is smaller in size as compared to the trophozoite. Right, that is 10 to 20 micrometer in the diameter. It is oval shaped with a blunt pseudopodium. Okay, then its food vacuoles will disappear, and it has the characteristics nucleus. Okay, now the third morphological form, that is the cyst. It is spherical. You can see the difference between the three morphological forms. These are very distinct, okay, and these are very different from each other. Then third one, the cyst, in that it is spherical. Then it is having 1 to 15 μm in the size, in the diameter. It is more smaller in the precyst. So the larger size trophozoite, then the precyst and the cyst. Alright, then it is uninucleatedly nucleated, binucleated and the tetranucleated. Cysts are present only in the lumen of the colon and in formed feces. Okay. Now we will study the life cycle of this parasite. Then in the life cycle, it is having only one host, and that is the man. In case of the malarial parasite will have the two hosts, mosquito and the human. Okay, mosquito plus the human. In this, in the Entamoeba histolytica, it will have only one host, and that is the human. Okay, now you can see in this picture, the human will consumed contaminated food or the contaminated water, right, where this trophozoites can remain, right, very well. Then due to this, they will enter into the human as the mature cyst. Now concentrate on the terms mature cyst forms it will enter, right. Now in that cyst, now what will happen, cyst is multinucleated, right, and therefore it is having more number of nucleus. Now excision will happen. Now one of the nucleus will get separated from this mature cyst. Okay, one trophozote with the 4 nuclei emerges, divide 3 times, and each nucleus divide once to produce 8 trophozoites from each cyst. Okay, which means one trophozote, you can see the 4 nucleus, that is called as the one trophozote. Okay, trophozoite, that will divide 3 times. Okay, after dividing 3 times, each nucleus from this 4 divide once to produce 8 trophozoites. You can see higher number of these trophozoites are producing by a single cycle, right. Therefore the 8 trophozoites from the each cyst. Okay, now after the excistation, one nuclei, one trophozoite will separate out. That trophozoite migrate to the large intestine. Now in case of the cyst, we have saw in the earlier slide, it will present in the colon, so it will present in the large intestine. Okay, now trophozoite multiply by the binary fission, it will divide. Okay, the 2 trophozoites will be formed, one of which will be for invasive infection and one will go for non-invasive infection. Okay, now we will see first invasive infection. Now that one trophozoite is entering into the inner side, invade into the intestinal mucosa and will cause the invasive infection. Invasive infection through the bloodstream, infecting sites such as the liver, brain and lungs. Okay, and the next trophozoite, in this the encystation will be happened. You can see in the starting there will be excistation, exit of the cyst. Okay, exit from the cyst, and here entry into the cyst, so it will called as the encystation. Then their maturation cycle will be takes place, that will result into the immature cyst. After immature cyst, then it will result into the quadrinucleate cyst. Okay, mature cyst, and which cyst exit into the host, exit in host via the stools, via the diarrhea. It will result into the diarrheal infection. Okay, and this non-invasive infection cyst will exclude from the body via the stools, via the feces. All right, and this is the life cycle of this Entamoeba histolytica. So we can say it is having only one host. First the mature cyst will enter. Mature cyst, one trophozoite will separate out. That trophozoite will then be divided into the 2 parts, invasive, non-invasive infection. Invasive infection will be in the liver, brain, lungs. Okay, then the trophozoite, one of the trophozoite by the binary fission will enter into the non-invasive infection. There it will again encystation will happen. After the encystation, immature cyst will then produce the mature cyst. Okay, chordinucleate cyst. Okay, and that cyst then exclude from the body via the stools. This is the life cycle of the entamoeba histolytica. Okay, then we will study the pathogenicity. In the pathogenicity, what is the mode of transmission? The mode of transmission will be the fecal oral route, right? Just now we studied the cyst will exclude from stool. So, transmission via fecal oral route by the uncontaminated contacts, improper hygiene, sanitization, and with ingestion of contaminated food, contaminated water. In this sites, this parasite can stay. Alright, therefore, by ingestion of contaminated food and drinking water, the parasite can get entered into the human body. Right? Now, first one is the intestinal amebiosis. Okay, in that, intestinal amebiasis indicate that organisms are confined to gastrointestinal tract. Incubation period is 1 to 4 weeks. Okay, it will what produce the intestinal amebiosis. This will be resulting due to the parasite entamoeba histolytica. In this, the incubation period is 1 to 4 weeks. In this period, they will multiply, right? They will again divide by the binary fission. Then again mature cyst will be produced and invasive infection and the non-invasive infection will be developed. Alright. Then the amoebae invade the colonic mucosa, right? Producing the characteristic ulcerative flask-shaped lesions and a profuse bloody diarrhea. Presence of blood in the stools. Alright. This is the infection caused by this parasite. Now we will see the symptoms. Now we have both type, mild symptoms, severe symptoms, right? In case of the mild symptoms, there will be the loose stools or the loose stool diarrhea. We can call that as the diarrhea, right? Including slimy diarrhea with pus, which is often foul smelling and painful passage of stools. Then stomach pain, stomach cramps will happen, then nausea will be there. These are the mild symptoms. Alright, now if we consider severe stage, then the severe symptoms will include amoebic dysentery. Okay, in this what will happen? In the stools the blood will be present, abdominal pain and the fever. Now profuse diarrhea. The patient may pass about 10 to 12 stools during an acute episode and still constantly feel an urgency to pass these stools. Okay. So the person will feel, alright. Then the profuse diarrhea, then the liver abscesses. Okay, then liver abscesses, severe ulcerations will be there, then severe gastric distension of the bowel, that is the stools, then peritonitis, that is inflammation of the intestinal wall and its lining. Okay, can be also called as the colitis, that is the colon, inflammation of the colons. Okay, then megacolon, this is very rare, in 0.5% of the cases it is happened. Then amoeboma, which result from the formation of annular colonic granulation tissue and may mimic the carcinoma of the colon. Okay, this symptoms, the second last and the last one, will result in some rare cases. All right, the rest are present in the severe symptoms and the above the mild symptoms. All right, these are the symptoms for this entamoeba histolytica infection. Now we will study the laboratory diagnosis. First, we have to collect sample. Now in this cases, we have to collect two samples. One for invasive infection, second for non-invasive infection. First, we will collect the blood sample. We will do blood examination. Then we will do the stool examination, right? Stool sample. Now in acute amoebiasis, stool or the colonic scraping from ulcered areas are examined by macroscopically and microscopically, right? Then in the blood examination also, it's shown the moderate leukocytosis. Now these are negatives in early case, however, the blood examinations. In later stages of the invasive intestinal amoebiasis, right, means if the stage when there will be the intestinal amoebiasis, that is invasive infection, then antibodies will appear and the serological test become positive. Okay, in the initial period the blood examination test can result the negative. After this some duration, okay, then the invasive infection will start proliferating, then antibodies will produced, there the serological test will be positive. Now this test will include the indirect heme agglutination antibodies, okay, then indirect fluorescent antibodies test and the ELISA test. With the help of this test, we can determine antibodies that are produced against this pathogen, against this parasite. Okay. Now, in the hepatic amoebiasis, the diagnostic aspiration is the trophozoites of the entamoeba histolytica may be demonstrated by microscopy. Microscopically, it can be demonstrated, right, with the help of the pus sample. Okay, that can be taken in the amoebic liver abscesses. This case is very rare, less than 15% of the cases. Then the liver biopsy sample can be used for the liver abscesses. Okay, then the blood examination and the stool examination, again in the blood type it shows the leukocytosis, okay, which means the killing of the blood cells, the leukocytosis, with the total leukocyte count of 15 to 30,000 microliters, which is 70 to 75% polymorphonuclear leukocytes are present due to the proliferation of the cyst inside the blood. Okay, then in the stool examination, in less than 15% of the cases, okay, these are examined, stool examination has been demonstrated. This indicates the persistence of intestinal infection. All right, now the serological test that we have studied earlier, we have saw the heme agglutination antibodies, then indirect fluorescent antibodies, ELISA, slide agglutination test, co-agglutination test. These tests are used to determine antibodies. Alright, this test, serologically test, are help to determine the antibodies produced against this parasite. Next molecular test that we can use is the PCR. Okay, then this is the laboratory diagnosis. Okay, in this importance is we have to do the two examination, blood examination and stool examinations. In the blood examination the leukocyte count will be low. Okay, in the stool examination there will be the amoebic hepatitis cyst. Okay, presence of cyst will be observed in the stool examinations. Okay, sample we can take blood sample, stool sample, scrapings of the ulcers that are produced in the intestines. Okay, then we can use the antibody test using the serology, and then we should use the molecular test such as the PCR and DNA probes we can use for determining the genome or to determining this parasite. Then in treatment of amoebiasis is based on the use of the amoebicides cidal drug, which means drugs that are responsible for killing this parasite. Now this with the luminal actions are the d-iodohydroxyquine, deloxynide furoate, and the paromycin. Then these drugs which are effective in the liver, intestinal wall and the other tissues. See, accordingly the drugs are available also for invasive infection and for non-invasive infection, right? For invasive ones, the amethine and the dihydroamethine drugs are available. Okay, and in the liver, chloroquine is only effective. Then in the tissues and in the intestinal lumen, the metronidazole and the nitroimidazole drugs are available. Okay, this infection is also totally recoverable as the drugs that are available, okay, against this infection. Then we will study the prophylaxis. In the prophylaxis, what measures or what prevention we should take to avoid this infection or to cure. Okay, then the amoebic infection, this amoebic infection can be prevented by avoiding the fecal contamination of food and the water. This is the foremost step we can do to avoid this infection, right? Because this infection is transmitted via the contaminated food, contaminated waters. So we should maintain proper hygiene. We should prevent the fecal contamination with the food and the water. Then there should be proper disposal of the human feces through the proper drainage system, right? Contamination can result from the sewage discharge into the rivers, into the open places. Then purified water should be distributed through the pipelines to avoid contamination. Through the pipes also, through the normal routes, okay, if you are taking the water from directly the river, then water may have contamination. To avoid that, the pipelines of the purified water should be applicable. Okay, and more than that, the boiled water is safest. Then amount of the chlorine normally used to purify this water is insufficient to kill cyst, and that is why the boiled water is more preferable. Okay, then vegetables that are usually eaten raw should be cleaned, or most probably that should be eaten boiled, or they cleaned with the uncontaminated running water and treated with the 5% acetic acid before its consumption. These are important steps, important measures that we should take to avoid this type of the infection, right? These are very easy ways we can do at our home, okay, that will help us to avoid this type of infection and the spreading of this parasite, Entamoeba histolytica. Right? Basically, we have to take care of the food we eating, the water we are drinking, consuming. We should not contaminate. Then the feces disposed should be proper. There should be not contaminated water consumption from river or any other source. There should be not sewage exposure into the rivers, right? Should be open places. That should not happen. Then water we are drinking should be boiled. Okay. This are the prophylaxis. This is the prophylaxis. So these are the measures, preventive measures one should apply to avoid this infection. In this online lecture, we studied the Entamoeba histolytica structure, Morphology, we studied three morphological forms: trophozoites, precyst, and the cyst. Okay, from that, then after that we studied the life cycle, right? Life cycle of this parasite. It is more, it is also present, its prevalence rate is also more. All right, then we studied the laboratory diagnosis, we studied the symptoms, the mild symptoms and the severe symptoms. Okay, then we studied the treatment, after treatment the prophylaxis and the preventions, right, measures. In case of this also, if we look into the geographical distribution of this entamoeba histolytica infection, it is worldwide available. Worldwide amebiosis, which causes the 40,000 to 100,000 deaths every year. So its prevalence rates is also very high, right? And the people who are staying near the infected or near the unhygiened areas, okay, near where the water is contaminated, water can contaminated, so that regions will be more at risk for getting this infection. Okay, therefore we have to take the proper measures, proper prevention to avoid this type of infection. I hope you understood this online lecture. If you have any doubt regarding this, please ask in the comment section. Thank you for watching.